Down at the microscopic level, there is a war that's been raging for billions of years. Bacteria are in this constant, relentless fight for survival. And it's not just against things like antibiotics, but against these tiny little genetic parasites that try to hijack them from the inside out. So today, we're going to dive deep into this ancient arms race and uncover a really brilliant, kind of sneaky defense strategy that's been hiding in the bacteria's own DNA all along. All right, so let's just jump right in with a pretty wild question. What if a parasite could actually make its host addicted to it? I know, sounds like something straight out of science fiction, but this is exactly what certain parasites called plasmids do. Now, plasmids are these little circular bits of DNA that live inside bacteria, and they have this incredibly clever and honestly kind of sinister trick to make sure they never ever get kicked out. The weapon they use for this is called a toxin-antitoxin system, or a TA system for short. The easiest way to think about it is like a two-part molecular switch. You've got a poison, the toxin, which is really stable and hangs around for a long time. And then you have an antidote, the antitoxin, which is super fragile and breaks down really quickly. The plasmid is constantly producing both of these, so, as long as that antidote is being made, the bacterium is totally fine. So how does this actually create an addiction? Let's walk through it. Imagine you've got a bacterium. It's got the plasmid inside, everything's cool. It's making both the poison and the antidote. But bacteria divide, right? They split into two new cells. Now, what happens if one of those daughter cells, just by chance, doesn't get a copy of the plasmid? Well, that U cell has lost the blueprint to make the antidote. And because that antidote is so fragile, the supply it had just disappears. Poof. But that stable, long-lasting poison, it's still hanging around. And with nothing to neutralize it, it kills the cell. This whole process has a name: post-segregational killing. So you see what's happening here, right? The plasmid has set up the perfect hostage situation. The only way for a bacterium and all its future generations to survive is to keep making that antidote. And to do that, they absolutely must keep the plasmid. They are, for all intents and purposes, addicted to their own parasite. And that's where this whole story took a really weird and mysterious turn. Scientists got how this worked for plasmids. It made sense. But then, they started looking at the bacteria's main DNA, its chromosome, and they found these exact same toxin-antitoxin systems hardwired directly into the bacteria themselves. This was a huge puzzle. Why on earth would a bacterium carry its own suicide system? And the mystery got even deeper when they started actually crunching the numbers. When they looked at common bacteria like E. coli, they found something really strange about where these systems were located. As you can see from this chart, they were way more common on the main chromosome than on the parasitic plasmids where they were first found. From an evolutionary standpoint, that just didn't make any sense at all. Think about it this way. It's kind of like finding a self-destruct button that's hardwired directly into your house's main electrical panel. It seems like a massive, unnecessary risk. Why would you ever want that there? It just seemed completely backwards. Unless, of course, it was there for a totally different reason. And this is where the clues really started to pile up. Let's look at the data. For one specific TA system, called REL-B, scientists scanned thousands and thousands of bacteria. And what they found was this incredibly striking pattern, which you can see right here. A given bacterium either had the REL-B system on its chromosome or on a plasmid, but almost never in both places at the same time. Okay, so was that just a fluke? A one-off? Well, they checked another system, this one called MASIF. And there it was again, the exact same pattern of mutual exclusivity. A bacterium had it on its chromosome, or it had it on a plasmid, but not both. And just to be absolutely certain, they looked at a third system, YFM/YOB. And, you guessed it, the pattern held. Thousands of data points, same result. This was no coincidence. This consistent, non-overlapping pattern had to mean something really important about what these systems on the chromosome were actually for. And the evidence was just overwhelming. We're talking across thousands of different bacterial strains, multiple different TS systems. And over 97% of the time, the pattern was the same. A system was either on the chromosome or a plasmid, but not both. This striking mutual exclusivity, that was the key that unlocked the entire mystery. Because here's what it all means. That TA system on the chromosome isn't a suicide button at all. It's a defense shield. It's an anti-addiction system. See, when a plasmid tries to use its toxin to hold the cell hostage, the identical system on the chromosome provides a steady supply of the antidote. This completely neutralizes the plasmid's threat and allows the bacterium to get rid of the plasmid without dying.
So the same weapon has two totally different functions, and it all depends on where it's located.
This whole idea became known as the anti-addiction hypothesis, and it was summed up perfectly by the researchers themselves. They said that these chromosomal systems act as a kind of immune system, protecting the host from the plasmid's nasty parting shot and allowing it to be cured. And this slide just nails the solution. That thing that looked for all the world like a dangerous self-destruct button was actually a secret eject button. It gives the bacterium a safe way to kick out a costly parasite that it doesn't want or need anymore. And this discovery revealed a much, much bigger story. This wasn't just about one clever trick. It was about a dynamic, ongoing evolutionary arms race between bacteria and their plasmids. This whole cycle, which they call the addictive selection model, explains how bacteria actually collect these eject buttons over millions of years. Let's break down how it works. Okay, so it all starts with a plasmid invading a bacterium and taking it hostage using that addiction trick. But then, every once in a while, through a random genetic event, that TA system actually jumps from the plasmid and gets copied into the bacteria's own main chromosome. And that is the absolute game changer. That bacterium now has its own permanent source of the antidote. It can safely eject the plasmid and survive. These newly immune bacteria thrive, and over vast stretches of evolutionary time, they basically collect an arsenal of different TA shields from all the different plasmids they've managed to defeat. So what's the big takeaway here? These genes that seemed so puzzling, they're not just random bits of code. They are a molecular memory. They're like battle scars or maybe trophies that record the entire history of a bacterium's ancient wars with its genetic parasites. They have literally shaped its genome. This incredible back and forth, this constant push and pull between parasite and host, has been going on for billions of years. It's a hidden war that has shaped life as we know it. And that leaves us with one last big thought. What other echoes of ancient conflicts, what other brilliant strategies and secret weapons are still out there waiting to be discovered, hidden in the DNA of all the life that surrounds us every single day?